like to say a special thanks to those of you who are with us from distant time zones. I know, for example, Paula Dawson Martins and Wits Lendra were up until 2 a.m. in the Netherlands, and actually even did some extra running of genomes through DRAM yesterday for some extra credit. So shout out to them and to all of you who are visiting us from far away. I made a Google Doc that contains a bunch of links, and thanks to Aditi Sengupta for the great suggestion in our feedback channel over on Discord. I will share the link of that Google Doc with you. If you want to check that out, it'll have a lot of links to some of the materials that were presented yesterday, and we'll continue compiling those links as we get through the presentations today and through the rest of the week. And there's also some general links there that give you some information on how to access KBase or how to get to the project web, the summer school website and so forth. So with that, I don't want to take too much of the time away from our presenters. Our first presentation is going to be given by James Stegen and Vanessa Garayburu-Caruso, and they are going to be talking about metabolomics in river corridors. And so James and Vanessa, please take it away.
Thanks a lot, Tim. Also, just a logistical thing. I think questions specifically come through the plenary session channel in Discord. Is that right, Tim?
That's correct. The plenary channel, and then there's also probably a lot of discussion in the Metabolomics channel today.
Great. Excellent. Yeah, welcome everybody to the second day of the EMSL Summer School. As Tim was saying, really appreciate everyone joining us. As Tim mentioned, today is about metabolomics in river corridors in particular. And I'll give you just an overview of what we're going to do in this particular talk, and then we'll move from there. So this talk is going to start with some overview thoughts on kind of orienting us to how we think about metabolomics, which is not necessarily in a very traditional way, a little bit different than you might be thinking about metabolomics. I'll describe that in the next couple of slides. I'll give a little bit of context such as that and some motivation elements. And then Vanessa, one of our colleagues here, is going to give you more of a science talk on how we've been looking at metabolomics and linking them to biogeochemical function. And then I'll come back just at the very end to give you an orientation to the rest of the day. There's been a couple changes in the schedule, and I'll detail those at the end there. So let's see. So with that, and we'll also have a few minutes of Q&A after the end of this presentation. So, okay. So a little context on terminology to start with. So as I mentioned, we're not taking necessarily a precise traditional approach to metabolomics. We take a very broad view. So traditionally, metabolomics, if you think about that, usually people think about small molecules, central metabolism, and sometimes metabolomics is split into those metabolites within as well as outside of cells. We take I guess a broader view on this. We're examining small molecules in environmental samples, generally almost always water-soluble organic molecules. And we conceptualize those as metabolites in the environment, and those are coming from diverse sources. They come from microbes, they come from plants, they come from other kinds of organisms. And we really think about them as the organic molecules that fuel as well as are produced by metabolic processes. And Vanessa, if you could go to the next slide, please. I should come through there. Yeah, so and from that perspective then, taking that broader view of metabolomics, and often what we're looking at is our secondary metabolites, but we don't quite parse it in terms of within and within cell versus extracellular metabolites. But generally, as I was saying, looking at water-soluble small molecules in the environment that are biogeochemically and metabolically relevant. And you'll hear a lot today about FTICR mass spectrometry. And this is one tool that we use in particular with respect to WONDERS, as people are aware. That's the sort of the data foundation for the summer school. So WONDERS has so far focused on FTICR mass spectrometry for doing metabolomics, and that stands for Fourier transform ion cyclotron resonance mass spectrometry. It's a long name. I'm not going to go over the technical details of FTICR at the moment. Will Q, during the virtual tour today, will give you a lot more technical information on the instrument itself, and Vanessa will go through more details in terms of what the data look like and how we're using them. But I want to really emphasize that this is just one tool in the toolbox. And by no means is it meant to be the only thing that we use. It's a great tool for doing untargeted analyses of complex environmental samples. It's very, very high mass resolution. We can assign molecular formula. We can do a lot of interesting things that you'll hear from Vanessa about once we can do that. And so, but I also want to point out that FTICR is not the silver bullet for metabolomics by any means. It's not quantitative. It does not give you concentrations. And it misses the very small molecules, many of those that would be traditionally associated with metabolomics, and in particular those molecules tied to central metabolism, such as glucose. And ideally, FTICR is paired with other methods, such as NMR, nuclear magnetic resonance, LCMS, or liquid chromatography mass spectrometry, gas chromatography mass spectrometry. Those are other methods that all have their own strengths and weaknesses, and as a collective can provide a broader view of metabolomics in environmental samples. And Will Q will also touch on those additional technologies, even though we won't be working with those kinds of data today during the summer school. So Vanessa, if we could go on. So that's a little bit of just orientation in terms of terminology and the instrument that we'll be focusing on today. I want to give you some broader conceptual context of why do we focus on, why are we interested in metabolomics from this particular point of view. And there's for a number of reasons, just a moment. So a number of reasons, and one of them has to do with the data when viewed in this way carries a lot of information with respect to microbiology as well as biogeochemistry. And this image here on the left is just an example of linking metabolomics data to microbial ecology. And I'll just briefly go over this, just touch on this, where the vertical axis here has to do with the strength of deterministic ecological selection that is imposed upon microbial communities. So as you go up the axis, selection becomes stronger. And the horizontal axis has to do with the thermodynamic favorability of the metabolites that are in those systems, in those samples. And you can see there's a relatively good relationship between the strength of selection that is driving the ecology of microbes and the thermodynamic properties of the metabolites that they are interacting with, both as resources and products of their metabolism. Vanessa, if you could advance there. And then in addition, the same horizontal axis having to do with the thermodynamic favorability, and this is something that Vanessa will tell you more technical details about, and Hyun Saab will go into even further details of the theory tied to the thermodynamics. And here we have the vertical axis being respiration rates, in this case aerobic respiration rates, being related to the thermodynamics. And so we see this over and over again in our work, where we look at metabolomics through this broad lens, we see it connected very strongly over to the ecology of microbes and emergent biogeochemical function. So, and Vanessa, if you could go to the next slide, please. We can go beyond that, linking metabolomics to other kinds of omics, and this is just a real, just sort of scratching the surface here. On the left-hand side, we have an ordination of mass spectrometry data, the FTICR data, the metabolomics data. Those are the circles, and then those are tied to metagenomic-based inferences of microbial taxonomy. Those are the arrows, and we can see some correspondence. And these are data coming from the WONDERS samples that you all have been working with yesterday, and we'll work with more today. So we can see correspondence between the microbial taxonomy and the metabolomic profiles. And on the right-hand side, we can generate functional hypotheses. And this heat map here, this is the 7 different rivers tied to the WONDERS sampling campaign that was described yesterday. And the colors have to do with the frequency of observations of different EC numbers that we're linking or inferring from the metabolomics data. And so you can start to get a sense of different rivers have different kinds of profiles, and we can generate functional hypotheses about what might be going on metabolically there from the metabolomics data. And that's work from Bob Danzak. You can speak to him in greater detail about that. In addition, we think a lot of our work is tied to understanding fundamentals, but also driving towards enhanced predictive capacity at a broad range of scales. And so another really interesting aspect of looking at metabolomics in this broad view is that we see real coherence in structural organization that in this case, as I'm showing here, is emerging at the continental scale. And we often think about metabolomics as, in small samples, in biological cultures and things like this. But if we view it this way, with broad sampling, this is tied to the summer sampling campaign that Amy Goldman mentioned yesterday very briefly. We can see emergence in structure of thermodynamic properties. Here on the right-hand side, the vertical axis is a thermodynamic parameter lambda that Hyun Saab will tell you much greater detail about what that is. Basically has to do with the efficiency of organic matter turning into microbial biomass. And so if we look at that bounding box on the left, those samples from the Wonders campaign, if we take those data, get the median lambda values from all the metabolomic samples and relate them to longitude, and we see a pretty darn good relationship there, considering that all those samples came from many diverse kinds of river systems with all kinds of different impacts. And so we see structure, and that's really encouraging in terms of being able to use the knowledge we gain from metabolomics to inform models, even working at continental or larger scales. In those 3 slides, I want to emphasize something here that we're showing you really the newest stuff. Okay, I don't know if people noticed, but I had on the lower right there, in prep. Those 3 slides are from papers, manuscripts that are in prep right now. And so we're opening the door here to a very active and dynamic and constantly changing research area. So we're not presenting like the most polished textbook kinds of materials to you today. Really, we're presenting to you the bleeding edge of what we're doing from a research perspective, which I hope you find to be encouraging and exciting to be kind of exposed to and engage with, and in turn advance those research domains. So Vanessa, if you could go to the next slide, I think that is the start of Vanessa's talk. So I'll leave it with Vanessa. I'll come back at the very end with that. Please, Vanessa, take it away.
Hi everybody, good morning. So I'm going to be telling you today the story about my newest paper. It's called Carbon Limitation Leads to Thermodynamic Regulation of Aerobic Metabolism. So first off, my name is Vanessa Garayburu-Caruso. Everyone calls me Vanessa GC or VGC, so feel free to do so as well. So these pictures are kind of a little bit about myself. I have a lot of experience doing lab tests and analytical instrumentation and field work, and today I'm just going to be telling you the story about my newest experiment. And so first, let's start with some background. So organic matter processes are key for riverine biogeochemical function. So within the river corridor, areas where groundwater and surface water mix, hyporheic zone, contribute significantly to aerobic respiration of the whole river ecosystem, contributing around 90% in some cases. So because of this, it is really important to understand what processes control this organic matter respiration in the hyporheic zone. There have been many biogeochemical models trying to integrate what we know so far into local scale reactive models to try to then understand the processes that govern respiration at a global scale. However, we currently have a knowledge gap. And the knowledge gap is driven because a lot of these models consider carbon just like a bulk pool and only represent very simple compounds that do not do justice at the variety and diversity of organic matter compounds that we can find in this hyporheic zone. So in order to bridge this gap, we need to have a better understanding of how carbon thermodynamics, concentrations, and the ratios between carbon and nitrogen are affecting this overall respiration rate. And so in order to do this, we have a great tool, which is the FTICR-MS that James mentioned in the introductory slides. So briefly, the way that it works is that you go to the river or you have your lab experiment and you take your sample. Then the sample is sent to EMSO for analysis. And the way that the data looks like, it's like this graph that I have here, where we have on the y-axis is an intensity of the peaks, and on the x-axis we have the mass of the compounds that are identified, or the M/C. these masses are extremely accurate. That's one of the greatest benefits of FTICR. And so because of this accuracy, we are able to determine that this molecule with this mass is precisely the one that we observe over here. So the way that we process the data is that after we have all of these spectra, which match them to a database, and we match these M over C's to a molecule with an error lower of 1 chemical shift. So with this information, then we can try to calculate thermodynamic properties of the carbon molecule. So one of the ways that we do this is by calculating the nominal oxidation state of carbon. The nominal oxidation state of carbon utilizes the number of atoms in the organic molecule that was identified to calculate just the oxidation state of the carbon. Then using an empirical relationship derived by Larroux and Van Capellen, we can utilize the NOS value associated to that identified molecular compound and then gather a Gibbs free energy. So what we notice is that these Gibbs free energies are positive. And the reason for this is because we need to pair them to an electron acceptor in order to make the overall chemical reaction negative and be favorable from a thermodynamic standpoint. However, in some cases, you have a dominant electron acceptor. In my case, would be oxygen because we're estimating aerobic relationships. So we can derive a lot of conclusions only on the Gibbs free energy of the carbon by itself, assuming there are constant electron acceptor conditions through the whole reaction. Once we have the Gibbs free energy, we can also estimate something that we call the biochemical transformations. So briefly, we have this compound over here that we showed in the previous graph, and then say we have this other peak that has an identified compound. We have a code that is calculating the mass difference between each of the peaks identified in a specific FTICR spectra, and then these peaks are mapped to a reference database, and depending on the difference, we can associate this difference to the gain or loss of a specific carbon molecule. So in this case, the difference between this peak and this peak is associated to the gain or loss of a valine molecule. And then depending on how many times these molecule transformation appear, you can also relate how many times this specific molecule could have been gained or lost within a specific sample. So some examples of where we have used these methods are in one of Emily Graham's studies in 2017, she observed by extracting the water-soluble fraction of riverine sediments that thermodynamically favorability was correlated with respiration rates, where thermodynamically favorable compounds were preferentially oxidized. In addition to this, Stegen et al. In a study in 2018 proposed a different conceptual model where carbon thermodynamics and concentration can be driving the respiration during groundwater and surface water mixing. So in his conceptual model, we have our groundwater in our system, which is aerobic, and it has more favorable organic carbon, but at lower concentration. And then we have the surface water in our system that has less favorable organic matter at higher concentration. And so the model suggests that when these two compartments mix, there is a combination between the concentrations and the thermodynamics that we are observing in the system that are driving the higher metabolic activity. So this graph over here is showing the distribution of carbon thermodynamics within our groundwater and surface water, where blue is completely surface water and purple is completely groundwater. Then we also have observed the importance of organic nitrogen cycling in hotspots. So a different study from Emily in 2018 has observed that reactions involving organic nitrogen were higher in areas that present also higher metabolic activity, while they were lower in areas of lower metabolic activity. This was also observed in a Stegen study where we have, I realized that the slide moved around, but where we have an overrepresentation of nitrogen-containing transformations in the hypereik zone versus nitrogen-free transformations in the river, being the hypereik zone the area of higher metabolism. So with this, the only thing we still have to understand is in a controlled lab experiment, how do carbon organic matter thermodynamics, concentration, and carbon and nitrogen ratios are influencing aerobic respiration rates. So for this, we conducted lab experiments where we collected sediments from the shallow hypereik zone in the Columbia River, and we took these sediments to the lab. We performed sequential extractions to induce carbon limitations. So this is a graph of the dissolved organic carbon in the extracted water for 50 hours until we induced carbon limitations, and we incubated these sediments. So our incubations consisted of 100 bioreactors where we had 4 treatments, 3 concentrations, and controls. And so in order to understand how the dissolved organic concentration was influencing respiration rates, we selected 3 different range of concentrations. The lower end mimics the concentrations we find in the groundwater in our system, while the higher range is similar to concentrations observed in more urban streams or streams with higher dissolved organic inputs. To understand carbon and nitrogen ratios, we selected model compounds that were either nitrogen-bearing or nitrogen-free. And lastly, to understand carbon thermodynamics, we selected compounds that mimic the extremes observed in our system, again, where blue is the surface water like thermodynamics, and the purple is the groundwater like thermodynamics. So we added all of these compounds into our bioreactors. This is how our bioreactors look like, and we incubated them while shaking for 6 hours at 21°C. We then estimated respiration rates as the dissolved oxygen decrease over time. So we measured dissolved oxygen non-invasively during the course of the incubation. So this is an example of how we did it. We had one dissolved oxygen dot and one pH dot, and with these fiber optic systems, we were able to estimate how dissolved oxygen change over time. And then we calculated the respiration rate as the slope in this regression line. So it is important to note, like I was mentioning in one of the previous slides, that our bioreactors stayed aerobic during the whole time. So in here, we can see that the initial dissolved oxygen concentration starts around 9.3 milligrams per liter. And by 6 hours of incubations, we have, excuse me, that the concentration ends in 3.7 milligrams per liter. Because of this aerobic constant environment, we are able to drive more conclusions on the carbon thermodynamic perspective. And so what we first observed is that respiration rates were limited by dissolved organic concentrations. So here we have on the y-axis the respiration rates, which is the slope of the dissolved oxygen decrease over time. And on the x-axis, we have the concentrations of our treatments. We observed that the respiration rates increase from 0.3 to 3, and then plateau from 3 to 9, suggesting that they were limited by dissolved organic concentration. So to dig in deeper into what the carbon character, the organic matter thermodynamics was telling us, we utilized the water, like the slurry water off of the incubation, and we analyze the FDICR data out of that post-incubation water. And so this is just like calling back to the methods um, we mentioned earlier in the talk. With the FDICR data and the molecular formulas identified, we calculate the nominal oxidation state of carbon and the Gibbs free energy of the carbon. And with this, we observe that with carbon limitation, respiration rates corresponded to organic matter thermodynamics. So this is the same graph that I showed you before. So on the y-axis, we have the respiration rates, and we have the carbon limitation zone is incubations of 0.3 and 3. And what we are observing is here on the y-axis, we have respiration rates, and we have on the x-axis, the Gibbs free energy of the carbon. And we see that under these carbon limitation conditions, more thermodynamically favorable carbon also has higher respiration rates. However, without carbon limitation, we did not see any influence of thermodynamic favorability in respiration rates. We see here are the respiration rates. Here's the thermodynamic Gibbs free energy. But without carbon limitations, we did observe that carbon and nitrogen ratios and nutrient availability explain respiration rates. So we observed that nitrogen-bearing incubations had a higher respiration rate than nitrogen-free incubations. So we took this to the next level and analyzed the biochemical transformations of all of our bioreactors. Once again, the biogeochemical transformations represent the potential carbon molecules that were gained or lost in a specific reaction and how many times this could have happened. And so what we observe is that without carbon limitation, the biochemical transformations shift with nutrient availability. So in this graph, we have an ordination plot where we can see the closer the points are together, the more similar those transformations are. And so for carbon limitation scenarios, we see that there is no real difference between the molecules that could have been gained or lost across both of our treatments. But when we overcome the carbon limitation, we see that there is a clear shift in the biochemical transformations that are represented in this reactor. So with all of this information overall, we can propose a new conceptual model. And in this conceptual model, both carbon, organic matter, thermodynamic, and nutrient limitation dually control respiration rates. So in this model, we propose a scenario where as you are initially, when you have low organic matter concentration, thermodynamic limitation controls respiration rates. And as this concentration increases, the thermodynamics become less important and nutrient limitations start controlling these respiration rates observed. And so this is important to include in biogeochemical models because these will help us better predict and under which scenarios these predictions apply when we try to incorporate higher resolution organic matter into biogeochemical models. And so with that, I would like to thank everyone that participated in this study, and also this was funded by Subsurface Biogeochemical Research, and the earlier plots showed by James are funded by Early Career Award. And I would like to open up for questions. On.
Yeah, so we can definitely do, no, that's great. We can stay on the previous slide. So I'll go over this kind of the logistical stuff right just a couple minutes before 9:15. So this is the time that if folks want to ask questions about the science, the concepts, what have you, we can dig in. I'm seeing some stuff in the chat as well as on Discord. Many of the questions there have already been answered, I believe. There was a question in the chat that I don't think has been answered yet, and it was let's see. So so this is one to me. So how do you calculate the lambda value that I presented in one of those slides, the one where it was linked to longitude. So that's really, I'm going to pass that question to Hyun Saab if he is available to speak at the moment, but I'll just say the short story is that lambda is tied to the stoichiometries of organic molecules as well as the metabolites, if you will. The elemental stoichiometries as well as their masses, and Hyun has developed a lot of theory around how to do that, to go from those stoichiometric ratios into that lambda parameter. And so Hyun, are you available to give just a really brief answer to that? I know you're going to go in much greater detail the rest of the day.
That is one of the topics that in my lecture in the afternoon, but briefly, the lambda is really calculated based on their energy balance between catabolism and anabolism. So we define metabolic reactions by combining catabolic and anabolic reactions such that their energy, for their energy balance. So lambda represents how many times catabolic reaction should run to provide the energy required for the synthesis of 1 carbon mole of biomass. So higher value of lambda means that so many times of catabolic reaction should run to provide energy, therefore thermodynamic efficient compounds. So I'll give more details later.
Thanks, Hyun. And one thing I'll say is that you'll be, there's R code, R scripts that are available to calculate lambda. And so that should be, I believe, Bob, that'll be part of Bob Danzak's tutorial. If not, it'll be part of Hyun's. So those kinds of scripts are available and can be, will be shared. So I'm seeing some other questions. Oh, actually, one thing I wanted to mention.
A bunch of questions over in Discord too. So do you want me to relay those some of those to you?
Sure. Can I just hit the ones that are in the Zoom for the moment, and then we'll jump to Discord?
Yep, let's do that.
Okay. Let's see. So one thing I'll say also with the values I was presenting on that slide, those are the median values of lambda. And so what that means is that every sample that you have a large number of lambda estimates. So every organic molecule, every metabolite that is identified in the mass spectra and that you can assign a formula to, we can then generate a lambda estimate. And so for every sample, you have a distribution of lambda values. And what I was presenting was the median for every sample. Okay, so some other questions here that I'm seeing. Let's see. How about someone asked, can you explain the significance of the hypereic zone? Vanessa, do you want to speak to that, please?
Yeah, so the hypereic zone has a different significance depending on the field that you are focused. But the short story is that it's very diverse in microbial communities. It has a lot of processing. It's considered a hotspot within the whole river corridor, and it is very rich in different nutrients. It has different redox ratios, redox conditions, and because of that, it's responsible of most of the processing that happens within a river corridor. And due to this higher processing, it is really important to study, because if you ignore it, you are ignoring the part that is responsible of most of the removal of contaminants, or most of the CO2 production due to aerobic respiration. And yeah, so it's a key ecological area to study for river corridor science.
Thanks, Vanessa. I'm seeing another one here. How do you disentangle thermodynamics versus nutrient limitation when organic carbon serves as the electron donor? I have some thoughts on that, but Vanessa, do you want to speak to that? I think it ties to your experiment.
Yeah, so the way that we were doing it over here was by looking at this relationship. So it was a very strong, nice relationship where we have respiration rates and thermodynamics correlated when there was carbon limitation. And these respiration rates within nitrogen-bearing and nitrogen-free treatments did not significantly change. And so when we overcome this limitation, we see that this relationship doesn't exist which is contrary to what we observed in the previous graphs. And we also observe that these respiration rates are different between the treatments on nitrogen bearing and nitrogen free. So for this particular experiment, this is the way that we are identifying where the dominance of nutrient limitation or thermodynamic limitation. So I'm thinking James really has like a bigger picture of how can we identify these in different systems that are not these precise experiments. So I can let him take on now.
Well, actually, what we're planning to do as we go forward in our research program is to build on Vanessa's experiment and to do additional manipulations using a finer gradient of organic matter concentration or DOC concentration, and evaluate the relative influences of thermodynamics and nutrient limitation across more of a continuum, if you will, to be able to develop, hopefully, like a functional form of the relationship between the influence of thermodynamics and organic matter concentration, and how that varies under different nitrogen contents of that organic matter. So kind of looking at the interaction between, like Vanessa was doing, but building on that, the interaction between the organic nitrogen and the organic matter concentrations using a more finely resolved experimental manipulations. So that's our particular approach, but I'm sure other people have additional ideas. So I'm seeing some other questions here. I went back to you, Vanessa. What are some potential applications of your research, of the outcome of your research?
So one of the applications that we had in mind actually when we were designing this experiment was for modeling. So we're hoping that by having specific scenarios where you can consider thermodynamic controls versus nutrient controls, you can expand the way that organic matter molecules are being represented in these biogeochemical models. So we are trying to build on the current and existing theory by introducing more substrate-specific types of models, which I think is something Hyun might talk a little bit more in his talk later on. And so we can consider millions and millions of carbon molecules, but by narrowing down the specifics at which we might have to drive these efforts in modeling in terms of thermodynamics or nutrient limitations, we can improve the predicting capacity of these models. And so this would be like one application that we were having in mind as we were designing this experiment, kind of trying to give scenarios that we can input into these models.
Great. I'm seeing some other questions here. How do you classify the compound classes, amino acids, sugars, using the FTICR mass spectrometer? So I'll take a crack at that, and others may want to jump in. But people have tried to do this using, often using a Van Crevelen analysis is what it's called. So you're basically taking the molecular formula and taking the stoichiometry of those elements and plotting them in a particular way. And then, so basically you have a two-dimensional space defined by those stoichiometries. And then there are some sort of general rules of thumb of where different kinds of molecules fall in that two-dimensional space. And that's this Van Crevelen analysis. You can just Google that. You can see some images of that. And people have developed basically different regions of that two-dimensional space that correspond to things like protein-like molecules or lipid-like molecules. And there's a number of other categories. I will say that we generally have moved away from that. Those designations are, I believe, initially developed from the petroleum industry, and it's not clear that they have really great relevance to natural environmental systems. And so we've moved more towards, in terms of classifying things, classifying them based off of their elemental composition. So do they have carbon, hydrogen, oxygen, or do they have carbon, hydrogen, oxygen and nitrogen. So sort of binning things based off of which elements exist in the molecular formula, as opposed to trying to define them as, protein-like, etc. I mean, there's still a lot of people do that. It's not clear that's so robust with respect to the FTICR in particular. Of course, other instruments can provide much more refined and clear identifications of specific molecules. And so the FTICR gives you the molecular formula, but it does not give you structure. And so that's kind of it's certainly a limitation of this particular methodology. Let's see. I'm getting another question here. Can you further explain the conceptual model? I think this is a question to you, Vanessa. How do we know when the model shifts from thermodynamic to concentration control?
So right now, we don't have a clear idea of how this curve looks like, because we only experiment with 3 concentrations. However, the study that James was mentioning is designed to provide a larger range of concentrations over in this axis and more spatial distribution, aiming to understand how this curve looks. Is it like a straight line? Is it an exponential function? So at the moment, we don't really know exactly when the shift occurs, just because we have limited information. However, we believe that this conceptual model will apply to also the basin scale samples that James was describing. And so kind of the idea that we had over here was that for example, headwater streams that had higher inputs of organic matter concentrations, respiration rate will mainly be limited by nutrient availability. And so these were represented over here by the yellow colors. And then as we move down in the basin, or maybe we have arid system or semi-arid systems that don't have as high organic matter inputs or dissolved organic carbon concentrations, we believe that the thermodynamics of the organic matter will be driving the respiration rates there. But the exact shift, it's still yet to come.
Okay, so I'm gonna cover a couple of questions I'm seeing, and then there's one more high-level question for you, Vanessa. So I'm seeing, can you tell us more about the FTICR mass technique? So you'll hear more about that. This is from Discord. You'll hear more about that from a Will Q in the virtual tour of EMSL. He'll provide you a lot of technical details. Someone is asking about the lambda calculation scripts. Those will be made available for sure. And let's see. So someone asked about what was meant by the term efficient or organic matter. Is it like carbon use efficiency? So lambda, I conceptualize that as something very conceptually similar to carbon use efficiency. How efficient is that organic matter or metabolites in terms of being turned into microbial biomass? So again, Hyun will dig into greater detail about that. So we have 1 more minute. And this question's for you, Vanessa. I don't think I asked this one yet. What is the meaning of delta G in the figure showing the relationship between concentration and thermodynamics? Is thermodynamics only determined by temperature? Can you say a little bit?
So delta G is the Gibbs free energy of the carbon at standard state conditions. And the way that it is determined, at least under this study, is only based on the nominal carbon oxidation state of carbon. Because we are calculating things at standard conditions, these numbers are relative to a 25 degrees C environment and a 1 bar atmosphere. And they just come from this empirical relationship derived by Larroux and Van Capellen. You can adjust these Gibbs free energy values based on the temperature of your study, and we are currently working on that. It goes parallel to some of the lambda development that we are currently working on, where we are adjusting the Gibbs free energy by temperature and pH. But the way that we have utilized it in this study and the previous studies that I mentioned in the introduction is just under standard state conditions. So we're assuming 25 C and 1 bar.
Okay, I think we're out of time. Thanks for the engaging discussions section. And let's see. Actually, Vanessa, will you pop to the last slide, please?
Yeah.
That's the. Let's see. We're almost there. A bit of lag. Okay. Okay. So the next thing up is Bob Danzak is going to go through the tutorial of the metabolomics analysis pipeline.